Hello everyone, I'm Ian Oldenburg, and I'll be telling you about our ongoing work using multiphoton holographic activation of cells to study the physical and feature-based microcircuitry of the visual cortex. It's often believed that cells in the cortex organize in functional groups to amplify and enhance visual signals. This architecture provides a convenient mechanism to explain visual phenomena like pattern completion and competition, and often the recurrent activity between layer 2-3 neurons is proposed to mediate this organization. The preferential like-to-like connectivity combined with a strong role of inhibition seems to support this theory. However, as cells that make up these groups are physically intermingled, it's very hard to study experimentally, and there are natural confounds in most experiments due to the feed-forward inputs from other layers and other brain areas, and we simply don't have enough information to know how or even if groups of cells coordinate to form these functional units. So the first challenge of this project was to figure out a way to actually study recurrent activity, and the coordination of multiple excitatory cells without the confounds that come from natural visual input. For this reason, we used a multiphoton holographic optogenetic approach that allows us to target individual cells without their neighbors. Specifically, we used the 3D shot approach with the chrome family of opsins. Using this approach, we can direct many cell-sized balls of light arbitrarily in V1 and drive excitatory cells with specific trains of activity. We expect that the general role for the cortex was to amplify signals. However, when we stimulated an arbitrary group of just 10 neurons with 10 spikes each in layer 2-3, most non-stimulated cells actually decrease their activity. So you can see here a targeted cell is reliably recruited across many trials. However, a non-targeted cell sees its activity drop below baseline. While in a given field of view, not all cells are suppressed, here are some that are activated and some that are suppressed, the majority are suppressed, and at the population level we see a decrease in activity that lasts for several seconds. Across hundreds of trials in many different mice and different fields of view, we see the vast majority of these ensembles evoke net suppression that follows a characteristic time course. This suppression, this mean suppression, depends on the number of spikes added and not on the rate or the number of cells that went into driving that effect. So if you add a few pulses into many cells or many pulses into a few cells, you get the same level of suppression. Seeing this overwhelmingly consistent suppression, we wondered what sorts of stimuli might actually break this blanket inhibition and drive net excitation. We reasoned that perhaps net activation can only be found if you stimulate a group of cells that would naturally be firing at the same time. We thus presented mice with different oriented gratings and programmatically selected groups of cells that were either co-tuned, such as down here, or not, such as in either of these cases. This, however, did not reveal any differences in the net effect of optogenetic stimulation, having a very consistent suppression across all of these variables. Even restricting our analysis just to the non-targeted cells that had the same visual preference as our stimulated ones didn't show net activation. However, if we instead look at the response of cells based on their distance to a stimulated ensemble, we see that nearby cells are consistently activated. These cells close by are definitely above zero, and this was one of the most consistent effects we saw across mice and things. We don't believe that this nearby activation is due to a light artifact, as this excitation is dependent on the entire ensemble that's driven, not just the closest cell, and persists even if you repeat the experiment in a set of mice that have a sparser opsin, in this case set by the CEP-W1 Cree line. Now, recasting our previous analyses, looking at those cells that are very similar, share a tuning with a co-tuned ensemble versus those that do not, we see that these nearest cells in these similarly tuned ensembles are excited, whereas the orthogonally tuned equivalent are suppressed. This supports the classic notion that similar cells are co-connected, but emphasizes that this effect only lasts for a few tens of milliseconds. We thought that perhaps by selecting cells that were close together, we might at last be able to overcome this inhibition and reveal net excitation. However, when when we do this, the net effect of a close-together ensemble of cells isn't more excitation, but is more suppression. And a consistent trend can be seen where, as you spread it out, you get actually less suppression. Finally, we wanted to look at how both the spatial and the tuning domains might interact. And so if you start by considering groups of cells, ensembles of cells that are untuned, if you put them close together or far apart, you don't get a very interesting, not a very dynamic difference. It's basically the same in both cases. But if instead you try to create an ensemble of close together co-tuned cells and compare that to an ensemble of far apart co-tuned cells, we have this dramatic difference, with the close together co-tuned cells being very suppressive, whereas much more so than we saw in an untuned group or anywhere else. And similarly, or conversely I should say, a far apart co-tuned cell drives an exquisite amount of nearby excitation, greater than you would get with an untuned group or in any other case. This indicates to us that there's a clear interaction between both location and tuning of stimulated cells, and the effects and how that affects the network. This further, however, indicates that there must be some tuned convergence in the excitatory to inhibitory network to be able to drive this special inhibition, and that this inhibition must follow feature selective rules. So with these peculiar experimental findings, finding that like-to-like connectivity only occurs at short spatial scales, but that tuned inhibitory connections dominate in many cases, we turned to our modeling colleagues to see if we could make a more complete model of cortical wiring that takes both of these spatial and tuning-based rules into account. We decided to design a spiking network model where the neurons in the recurrent layer receive their input from their neighbors based on both spatial and tuning-based rules. At the end of the day, in this model, a cell's final tuning preference is dictated by its final inputs. Nothing has a predetermined set tuning preference. And using this model, we can test out different spatial and tuning-based connection functions, as well as how many synapses are needed to be involved to match our experimental results. When properly tuned, we find that we can recapitulate our experimental results, but only when we include rules including a very small spatial scale connectivity and have some tuning preference in the inhibitory cells. We're now using this model to make and then test predictions about how other cell types will react and how other arrangements of excitatory neurons affect each other. With that, I'd just like to conclude. We observed that optogenetic activation of a small number of cells in layer 2-3 recruits mean suppression. Some cells are some activated cells are found nearby, especially when they're similar to the driven ensemble, but dissimilar cells are very inhibited. Putting many cells close together doesn't overcome inhibition, but instead amplifies it, an effect that's exaggerated following tuning rules. This provides evidence that there is feature-specific ensemble-to-ensemble connections, but inhibition is the dominant force at these scales. This work is a collaborative effort between the Doiron and the Adesnik labs and led by 3 postdocs, Will Hendricks, Greg Handy, and myself, with the support of members of both labs. We'd like to thank our funding sources and you for listening. Before you go, I'm starting my own independent lab at Rutgers this spring. If you're interested in using advanced multiphoton optogenetics, please reach out. I'd love to talk to you.